Thank you for viewing the Kineta Incorporated corporate presentation at the Master Investor Conference. Please also note that the company has a virtual booth at this conference that we'd welcome you to visit. My name is Sean Donato, and I'm a founder and the CEO of Kineta. For those of you viewing this presentation virtually, please make a note of the company's safe harbor and non-solicitation statements. Kineta is a Seattle-based biotechnology company. Our team is developing new immunotherapies that address critical patient needs in immuno-oncology, neuroscience, and neuropathic pain. We do this by focusing on a largely untapped area of immunology called the innate immune system. Our company's science has been validated by two key strategic partnerships with established global pharmaceutical companies. Kineta has a strong current financial position, and our team expects to achieve significant near-term milestones that will drive corporate value over the next 24 months. We are an experienced startup team with a goal to bring the company to an exit through an IPO or acquisition next year. Kineta's product portfolio is composed of novel drug candidates in oncology, neuroscience, and biodefense. During this presentation, we'll primarily discuss Kineta's development programs in immuno-oncology, including a novel drug candidate that blocks VISTA, a target involved in tumor resistance to the immune system. In addition, we'll talk about a program we partnered with Genentech involving the novel drug candidate KCP506. KCP506 is a first-in-class non-opioid treatment for chronic neuropathic pain. The markets for our products include two of the largest and most rapidly expanding commercial pharmaceutical markets, immuno-oncology and pain. The immuno-oncology market is currently an $88 billion market and is expected to grow to more than $150 billion by 2024. The neuropathic pain market is currently $6 billion and is expected to grow to more than $10 billion by 2024. These markets are also two of the most active biotech sectors for new investment, M&A activity, and initial public offerings over the past 3 years. Now, as a company, our principal focus is novel immunotherapies to treat cancer. Every year in the United States, there are an estimated 1.8 million newly diagnosed cases of cancer, and more than 6,000 patients die of cancer-related disease. In fact, cancer is the second leading cause of death in the U.S. behind heart disease. With over 20 million people living with a cancer diagnosis, there is a profound need for new therapies to improve cancer treatment. Our approach to cancer is differentiated from many other companies in the cancer immunotherapy space by our focus on a branch of immunity called the innate immune response. When most of us think of the immune system, we think of the adaptive immune response. The adaptive immune response is the specific immune response to a pathogen or vaccine that emerges slowly after infection or vaccination. The innate immune response emerges much earlier, within hours after infection. Innate immunity can broadly fight cancer and infection, and a strong innate immune response is important to drive a strong adaptive immune response. The innate immune system can also be either increasing or decreasing inflammation, and its programming helps to determine whether a tumor is inflamed or hot, as opposed to being uninflamed or cold. In cancer, Kineta is differentiated from other companies because we develop drugs that target the innate immune system to increase tumor inflammation and turn colder tumors hot. Our approach to reprogramming and increasing inflammation in the tumor microenvironment involves an important innate immune switch called VISTA. VISTA is involved in setting the tumor temperature to cold by reducing tumor inflammation. Kineta is developing drug candidates that block VISTA and, as a result, increase tumor inflammation and reset the tumor environment from cold to hot. So why is this important? Well, when we think about conventional treatments for cancer, in particularly advanced or metastatic cancer, we typically think of chemotherapy, surgery, and radiation therapy. When cancer is detected early, these conventional approaches are often adequate to achieve complete remission or a long-lasting response. However, in advanced cancers, conventional treatments can delay progression of cancer and can increase survival, but they rarely provide a cure. One of the reasons a cure has been so elusive is that the immune system does a poor job of developing an adaptive immune response to cancer. Over the past 5 to 7 years, we've seen the development of a novel class of drugs called checkpoint inhibitors. These checkpoint inhibitors can enhance the adaptive immune response in hotter tumors. So in patients that have a hot tumor, checkpoint inhibitors can result in a dramatic cancer regression with sometimes long-lasting results. Unfortunately, most tumors are cold and do not respond to checkpoint inhibitors. Because VISTA is involved in setting the temperature of tumors, its expression is associated with colder tumors and poorer survival in cancer patients. Kineta is developing therapies that block the action of VISTA and turn cold tumors hot. In colder tumors, the tumor environment is composed of many infiltrating innate immune cells, shown in blue, that prevent inflammation and block the activity of anti-cancer T cells, shown in red. The immunosuppressive innate immune cells have a lot of VISTA on their surface. Kineta has developed a VISTA blocking therapy that reduces the number of immunosuppressive innate immune cells and increases the number of inflammatory cells in the tumor, including anti-cancer T cells. As the composition of the tumor environment changes, an adaptive immune response to tumor emerges, helping fight cancer. By turning these cold tumors hot, VISTA blocking therapy should also increase the responsiveness to first-generation checkpoint inhibitor therapies. Shown here are examples of VISTA blocking therapy used in 2 mouse tumor models, both alone and in combination with a checkpoint inhibitor. As you can see in the graph on the left, which shows tumor size over time for a melanoma tumor model, single-agent anti-VISTA treatment results in a significant reduction in the rate of tumor growth compared to placebo. For example, compare the red diamonds of anti-VISTA treatment to the black circles of placebo. In the graphs on the right, we're testing the ability of VISTA blocking therapy to enhance the activity of 2 checkpoint inhibitors called CTLA-4 or PD-L1. In these examples involving a colon carcinoma tumor model, single-agent checkpoint inhibitor therapy, shown by blue squares, is less effective when used alone than when combined with an anti-VISTA therapy, shown by red triangles. The combination results in a nearly complete inhibition of tumor growth. Kineta is one of a small number of companies globally that are focused on developing VISTA blocking therapies. Most of these companies are in preclinical development like Kineta, but one company has an early phase 1 clinical product. Kineta believes that it has solved many of the fundamental problems facing our competitors, and therefore our VISTA blocking therapy has the potential to be a best-in-class treatment for colder tumors. So to summarize, Kineta is targeting VISTA to reprogram the tumor microenvironment and turn cold tumors hot. Since VISTA expression correlates with poor overall survival, we believe that VISTA blocking therapy can improve patient survival in a number of hard-to-treat cancers. Kineta is developing best-in-class immunotherapies with single-agent activity that also work effectively in combination with checkpoint inhibitors. The potential of our product is reflected in anticipated peak year sales of more than $3.5 billion in an annual addressable market of more than 500,000 patients. In addition to the work that our company is doing in cancer therapy, we are also developing a novel non-opioid treatment for chronic neuropathic pain. Our lead candidate, KCP506, is currently in phase 1 clinical trials in Europe. The problem of pain is the problem of overuse and addiction to prescription opioid products. Every day in the United States, more than 130 Americans die of accidental prescription opioid overdose relating to the overprescribing and illicit use of these drugs. Kineta's novel approach to treating chronic neuropathic pain involves the development of KCP506, an inhibitor of a new drug target called the alpha-9 alpha-10 nicotinic acetylcholine receptor. This receptor is involved in both pain sensation and inflammation around injured nerves. What's most exciting about KCP506 is that by blocking its receptor, the drug demonstrates analgesic, anti-inflammatory, and neuroprotective effects. KCP506 is also a non-opioid product. The drug doesn't enter the central nervous system, the brain, and spinal cord, and therefore does not have any of the negative neurological effects of opioids, including impaired cognition, coordination, and respiration. Kineta is the only company globally that is developing a drug with this particular mechanism of action. The uniqueness of our product can be readily observed in animal nerve injury models involving the spinal nerves. In this slide, we're showing a cross-section of the sciatic nerve. The sciatic nerve injury is responsible for many cases of chronic lower back pain in the U.S. In the sciatic nerve of normal animals shown in the cross-section on the right, you have a healthy bundle of nerve fibers similar to a fiber optic cable. Each of these fibers transmits a signal through the spinal cord to the brain. In injured sciatic nerve shown in the middle panel, the nerve begins to deteriorate over time through a process of ongoing inflammation and nerve injury. This is one way that an acute injury can lead to chronic pain through an ongoing process of nerve degeneration and inappropriate nerve signaling. Now, the animal on the right had the same sciatic nerve injury as the middle animal, but this animal received 2 weeks of therapy with KCP506. As you can see, the Kineta drug blunts ongoing inflammation and neurodegeneration in this animal, leading to a healthier looking nerve with less long-term pain. Because of the innovative nature of our pain program, Kineta was able to ink a major pharmaceutical deal involving this product with Genentech Roche in 2018. The partnership is structured as an option and license agreement. So far, Kineta has received $21 million in upfront and milestone payments, and we are eligible to receive a total of more than $350 million in development, regulatory, and sales milestones. Genentech has an option to take control of the development program after the completion of Phase 1, and Kineta is eligible to receive high single-digit to low double-digit royalties on the net sales of products that result from the partnership. Now, our team is composed of experienced biotech company founders and entrepreneurs, veterans of the pharmaceutical industry, and experienced finance, HR, and legal team, setting the groundwork for a solid exit for our investors. In addition, this team is supported by a science advisory board composed of world-recognized experts in immuno-oncology from a diverse group of prestigious academic institutions and hospitals. Over the last 3 years, we've seen a steady increase in the number of both follow-on offerings and IPOs involving biotech companies in the capital markets. In 2020, there have been over 60 biotech IPO and 62 licensing transactions over $50 million. While much of the broader market has suffered in a COVID-related downturn, the biotech sector has remained strong. Kineta is currently preparing for a potential public offering as early as 2021. The company has a number of important development catalysts across its various products and partnerships that we believe will drive value and investor interest over the next several years, including the continued development and regulatory approval to market both the VISTA-blocking therapy and our non-opioid chronic pain therapy. The tempo of these catalysts should position our company for a number of possible exits over the next 2 years. So in summary, Kineta is developing novel, best-in-class products for 2 of the largest global therapeutics markets, oncology and pain. We are an experienced management team that has brought previous biotech companies to a successful exit, and we're supported by a world-class science advisory board. If you would like additional information on our company, please reach out to the company through one of the representatives listed on this slide. You can also see us in the virtual booth at this conference, or link up with us through Facebook, Twitter, or LinkedIn. Thank you, and we look forward to keeping you updated on our continued progress. For those of you joining us from the Master Investor Conference, we'd like to give you a tour of our facilities here in Seattle, Washington. Leading the tour of our biotechnology facilities will be our Senior Vice President of Immuno-Oncology, Thierry Guilledeux.
That's where we are doing the flow cytometry analysis to detect the different marker on our cell line or directly in the blood. So we have 2 main machines here. It's the flow cytometer where we can analyze all our cells. So in this chemical we have 2 different areas. Here it's the molecular biology where we are preparing our clones and evaluate them also by PCR. And on this other side it's an octet machine where we can evaluate the affinity and the quality of our antibody that we are producing. And also the western blot area where we can see if our protein are well produced and stable. So in this chemical we are doing mostly ELISA analysis, so that's where we set up our plate for ELISA, and this the reader to analyze our ELISA luminescence. So in this chemical we are doing the biochemical analysis, where after producing our antibody we purify them and evaluate their quality in HPLC or mass spec analysis. So cell culture is a very important step in all our R&D work, and we have two chemicals to cell culture room, and this is the first one. Well, actually you have two classical hood and incubators. So this is the second cell culture cubicle, where we have three hood, a big one, and an incubator also here in the back.
Hi, this is Craig Phillips, President at Kineta. I hope you enjoyed your virtual tour of the laboratory today with Terry Guillaudot, and that he was able to share with you a little bit about what some of our research capabilities are here at our Seattle labs. We look forward to speaking with you more, and if you have any questions, please feel free to give us a call. Thanks very much.